So hi, hello everyone again. Hello and welcome. Microbehunter here. Welcome to another Saturday live stream, Saturday microscopy live stream. Hello all around hello to all around the world. So here we go. Yeah, today I want to show you again a couple of samples from a slide box. As a matter of fact, I've got 1, 2, 3, 4 slide boxes here, and also some mystery slides. Look at those here. It's in mystery slides because I am not able to read the slide label, but I'm going to show you a way of how we're able to automatically get this translated. And we're going to have a little guessing game as well. If you're new to this live stream, then of course, welcome. I've been doing this now for a couple of months, doing a weekly microscopy live stream to create a little bit of a microscopy community. And until everyone's joined in, I usually, it has become tradition that you quickly give me a feedback on how the sound quality is like, because every time when I start the live stream, I have to readjust the sound, and sometimes it's too loud and sometimes it's a little bit too soft. And also, if you are able to say hello in the chat, then I want to say hello back to the whole world, and I'm usually using the first couple of minutes to simply read off from where you are. So hello from Lebanon, hello from Vietnam, from Indiana, okay, and from the UK, from Hungary, from Netherlands, from England, again a hello from Vietnam, and so there are. Let me see here. I am excited for this one. Yeah, I'll be showing you a couple of slides. During the last couple of live streams, I've prepared a few slides. I can do this today as well, but today I decided again to simply show you a couple of readily commercial slides. Okay. We're going to be detectives. Yep, we're gonna have a look at some of those slides here. Hello from Utah. Okay. The sound is good. That's always good to hear. Okay, great. Okay, hello from France. Let's play detectives today. Okay. Hello from India, from Massachusetts, the United States, from Aargau. Okay, which is in Germany. I still remember the Rotterdam egg. Yep. And from the US, West Coast. Okay, and greetings from Finland. Okay, from Finland. I don't remember that we've had anyone from Finland up to this point. Hello in any case. Does anyone want me to take my microscope and look at a few samples from the slide box top? You're sure you can do that, but I think it might be a little bit difficult to share the pictures that you can take, because unfortunately YouTube does not yet allow you to share images in a chat. So what I'm going to do today is the following. Before I show you a couple of these slides that I've got here, and before we're going to have a look at those mystery slides, I want to show you a little bit of an experiment that I'm doing right now, which is not yet successful, okay? But I just want to explain it to you, and so maybe if you want to try it yourself, maybe you're a little bit more successful. And because I want to put pollen under the microscope, and I want to grow pollen tubes, and I want to show you what I've made here. So that's basically a little bit. I was kind of hoping to be able to show you the results, but I do not have any results. I'm just going to show you a little bit of what I want to do. So what I've got here is I've got here, cover glass here, and I've got pollen from a dandelion. Here, look, it's springtime here in Europe, where I'm from. That's a little bit of dried up dandelion, and I dipped the dandelion on sugar water. So I've got here 50%, 25%, 12%, 6%, 3%. So I always was having the sugar concentration, and by dipping I took actually not the whole thing here, but obviously I ripped out using some tweezers some of the answers with the pollen, and I dipped it into the sugar solution, hoping to be able to make the pollen germinate. Okay, it didn't germinate yet, so I'm going to still wait until tomorrow, and if I'm able to get this somehow optimized, and if I'm able to get it to work, then maybe next week or so I'm gonna actually show it to you. But it's not such an easy thing, and I'm going to simply show you what you're able to see here. So you're now able to see the microscope view. By the way, maybe you've already noticed that the layout is a little bit different. I've now basically changed to the layout again, and when you actually look here, and all of those little dots that you see, I need to go up with the magnification here, are the individual pollen grains. Okay, there were not so many. I actually also had when I tried this before, there were also more. but you do see that there are pollen grains visible. Okay, let me change this around here. So here they are. They kind of look yellowish, yeah. And those pollen grains, when they land on another plant, what they will start to grow a little pollen tube. It's really a tube that starts to grow, and this tube then contains the sperm for fertilization. And if you add sugar water of the right concentration, then and if you wait, and if everything's right, then you're able to see that the pollen grains start to grow. And this is basically something that I tried to do, but they don't grow yet. So I've been waiting now 2 hours, and I've tried different sugar concentrations, of course. So I hope that I'm able to optimize it. Let's try a lower concentration here of sugar. That's only a sweet percentage. I need to dry it off a little bit. Okay, and yeah, let's see if we're, yeah, even here, you can see at the lower sugar concentration, we're not yet successful, but maybe I'm able to find something here. Yeah, let's go up with the magnification a little bit. That's an air bubble. Yeah, yeah. Just wanted to share with you a little bit of what I'm currently working on. Yeah. Let's go up yet more with it's 40 times. Yeah. That's an individual dandelion pollen grain, and what we should actually expect if the pollen tube starts to grow is a little extension and really a tube growing from the pollen grain. And so far I've not managed to do this yet. I need to get the right sugar concentration. Maybe I have to add some other chemicals like there are certain elements, trace elements that are also needed. But I simply wanted to share this idea with you, and if you want during springtime to experiment around, if you've got some plants somewhere that have a lot of pollen, then you also might want to make a series of different sugar concentrations, add a little bit of sugar water, and then see if it will start to germinate. Okay, so I just wanted to share this with you, that this is one of the projects that I'm currently working on. Okay, but as I said, today I actually want to look a little bit more at some readily prepared slides. And what I would like to do, for those of you who are also new, you can post, of course, comments and questions into the chat box on the side. What I will do is that later on I will go through your comments and questions, and I will try to respond to some of them, even if they're a little bit off topic. As long as they're microscopy related, that's fine. Okay. If they're too far off topic, or if I do not know a meaningful answer, then of course I'm going to skip over it. Okay. So let's go back to the desk view here. Okay. Yeah. So I'm reading again. Hello from Ontario, Canada. Okay. You said that your birthday will be in May. Can you tell me when specifically, so that maybe you can do a live stream for your birthday? Ooh, I don't know if I have exact time on that, but I've got birthday on May 12th. Okay. Hello from Northern Germany. Okay. Hadre Miesenberg, we're going to the detectives later. Oh, okay, yes. So let's do the following. Here are some Chinese slides. Why do I have them, you might wonder. And I just want to tell you that this slide box was, I don't even remember which microscope it was, but it came along with a microscope. And this is basically what I got here. And I have to tell you, these are very standard slides. So actually I was able to identify I think most of them, even without translating the label. Yeah, we're just going to have a look at them in any case. And look, I've got here a mobile phone, and this mobile phone, I have got this Google Translate app, where I'm able to take a picture of this, and then it's going to translate this. But let's have a look at this at the first one first, and then I'm going to show you some other slides as well. And then we're going to also do a little bit of a slide quality comparison, because you can see that not all slides are equal. So some slides are actually of not so good quality. This one is a really easy one. You might already be able to find out what it is simply by looking at it without a microscope. We have to use a low magnification here. Yeah, and let's have a look. Oh, of course, I have to switch over to the microscope view. Here we go. Yeah, and here we are. Okay. Yeah, this is of course very, very easy. Look. Okay. If you want to, of course you can write the answer directly in the chat box on the side. Yeah. An insect leg, of course, right? But which one? We're going to find out soon. Now, one of the things that I want to mention here as well is which talks a little bit about the quality of the slide. Look at all of the dirt that you have here in the background. Of course, no slide is completely free of dirt and dust, but yeah, those here are indeed a little bit cheaper and not quite as clean. Yeah, and sometimes the dust is not on the slide, but actually inside, beneath the cover glass. So there's no possibility to actually also remove it. What I'm going to do now is I'm going to simply remove this filter. So I'm going to have it in bright field, so that in other words, so just like you would be able to see it. Okay. So of course, yeah, from an insect, your honeybee. Could it be a honeybee? Most likely it is. We're going to see. And one of the nice things about those prepared specimens is that generally the companies, ideally, they already use, of course, mounting media, which are very suitable. So, for example, normally when you put an insect leg directly under the microscope, they turn out to be extremely dark. If you also watch some of my previous live streams, the so-called clearing of insect parts is very important. So the correct mounting medium or correct processing, sometimes by placing the insect parts into potassium hydroxide, will actually clear the exoskeleton and make it more transparent. And this is something that's actually also necessary here. And yeah, generally, I don't know what this very dark part over here is. It is, yeah. So that's of course the claw of the insect. The question is now, which insect is it? And we're going to see if Google Translate is able to help me here, because I don't know Chinese, of course. Okay, could it be a fly? Well, we're gonna see. Okay, what I have now is I'm going to now change over to the desk view again, and here you have my my mobile phone. This is the Google Translate app. Okay, it's set to German right now, but that's okay. I've got here Chinese. I don't know if you're able to read it, but from Chinese to English. And what I have to do is I have to click the picture here, and I'm able to take a photograph. And when it takes a photograph, to focus here, when I click the picture here, not the picture, the release, then what does it say? I don't know if you're able to read it. B3rd film loading. Test 008. What in the world does this mean? Okay, that's one of the funny things that sometimes happens, that you cannot see it properly, unfortunately. Okay, but let's try it again, because it's with the exposure time. But it will actually say something here like B3rd film loading. That's really strange. Chinese language is a little bit ambiguous and very context dependent, but it does seem to be maybe the third leg or third leg of a bee. Okay, with the film loading, I don't know. Sometimes it will actually, if you try it again, produce a slightly different result. Okay, I have to focus all the time again. Let's try it again. Click. Yeah, it's still, now what does it say here? Now we've got more information. Honeybee, third pair of thoracic legs. That's already pretty informative. Okay, it's clear that it's got to be a thoracic leg, because that's where the legs are attached. In an insect, the legs always are attached to the thorax, which is the chest, right? But the film loading, I have no idea. Okay, maybe whole mount, could that be? Yeah, that whole mount maybe is translated as film loading. I don't know. Yeah. I think we can confirm that this is a honeybee. Okay, and it's the third pair of legs. It's interesting, if you retry to get it translated, sometimes you get different different answers. Okay, yeah. So this is already quite interesting here. So maybe I'm going to relabel the slides here for the future. Okay. Wow, there are lots of comments already here. Okay. So I'm going to go again through the comments a little bit. Can you explain what is exactly DNA sequencing? Maybe I'll do this a little bit later, but remind me, okay? Actually, or yeah, I'll quickly explain it later. But I think this actually is the info for a whole separate live stream. I have some confusion on the reproduction of unicellular ciliates. It is called binary fission, right? I want to understand the process. Ooh, wow. Oh, that's another whole. Thank you for the question, but that's a long one. Okay, so basically cell division. So I have to remind that I want to talk about later on about DNA sequencing. I want to talk a little bit about the division of ciliates. It's the leg of an insect. Can you show how tumors look in prepared slides later on? I have some medical slides. I always have had hard time with since I'm not familiar with medical stuff. Okay, I'm going to respond to that quite right now. I don't have slides, medical slides with tumors. However, I was able to once see one, and I was then a doctor explained the slides to me, and he said that's a tumor. And I asked, well, how do you know? And he says, yeah, one of the characteristics, I'm not going to quote, okay, I'm not a doctor, okay, but he said, yeah, because the cells look so extremely irregular. Okay, so this was actually the way that he was able to identify that it's a tumor because the cells in the tissue were very irregular, and this was somehow a sign of uncontrolled cell division. Okay, so I'm just passing on to you the information that I received myself. Okay. Yeah, the layout of your screen in this stream is well organized and a great improvement. Yes, the reason why I changed the layout again a little bit is it's a little bit less cluttered, and you're again a little bit able to see a little bit more here. And yeah, so that's simply the thing here. Okay. Yeah, there is a claw at the end. Yeah, and what do we expect since the sample is made in China? Always get some extra gift unknown particles with a sample. Let's put it this way. Dust can never be entirely avoided, okay? And sometimes if the slides are made very quickly, yeah, then of course you can, or not properly cleaned, then you're also able to get some other stuff on here. So this one here, I don't know what this is. I barely am able to see it, okay? So let's have a look at it as well, okay? And yeah, here we go again. And some scratches here, and I don't know. I'm starting off with a high magnification already, which is already a bad idea because you should always start off at lower magnification. Now I have to find it somehow wherever it is. You know, that's certainly not the thing I want to look at. Okay, so let's go further down with the magnification. So it's not the lowest; it's 4 times. Ah, here it is. Okay. I mean, yeah, let's go up a little bit here. So that's, I'm going to later show you some nicer examples, okay? A very, very typical, yeah, common thing here. I think it's cut too thinly, okay? Yeah. You are able to see the individual cells. Ooh, I just realized one thing. Normally I have this arrow that I'm able to use, but I forgot now. When I redesigned the whole layout of the screen, I forgot to include the arrow. Okay. So you know what? I'm going to try to do this. I'm going to, while you look at this, let me try to add it. Okay. Give me a second. Maybe I'm able to find the arrow. Here it is. Oh, okay. Bloop. Ooh, that's too big. I need to make it smaller. Okay. Yes, here we go. Because this arrow is a superimposed graphic, right? Yeah. Look at all these are vast so-called. Yeah, I'm not going to tell you yet, but obviously those little things that you see here, these are individual cells here as well, right? Okay, so what could it be? Yeah, it looks like a plant stem. Yes, of course it is. It's a plant stem cross-section, but the question is which type of plant. Now, I don't know which species it is, but it's very clear that it is a so-called a dicot plant because these vascular bundles are arranged in a circle. A dicot are those plants that have the first 2 leaves. Grass, for example, and wheat and grain and rice when the first leaf comes out, it's one leaf, or corn. There's one, so these are monocots, and dicots are all of the other plants that basically have where the first leaves are two leaves, right? We're going to have now a look at the, you know what, I'm going to go up yet again with the magnification here, and yeah. This is, I think, a typical example of a specimen that has been cut too thinly. I think the sample is too thin. Yeah. So you see, when a focus then immediately goes into focus and right away out of focus, and this is a sign that yeah, the specimen itself is not really very thick. Yeah. In that sense, it's yeah. I'm going to show you later some much nicer examples. Let's have a look here again. Let's use the camera again, and I need to have a look here at, to focus first. Picture. Dicot stem crosscut. Okay, it says here a dicotyledon stem. Yeah, as I told you, and it's a cross section. Yeah, so in that sense, I think yeah, the labels definitely match the what we expected it to be. Yeah, it's a stem of a plant, and it's a dicot. These are the plants that have first two leaves. Yeah, so this is yeah, another one that we finished now. Okay, maybe Aristolochia. Somebody just photographer 85. No, yeah, maybe, I don't know. But I've got some really nice specimens of Aristolochia cross sections here. This one is a very common one. I already knew it right away, just even just by looking at it, even without a magnification. It's a very common one that's often included, and I also made videos and live streams about that before, because it looks generally quite nice. Okay, and what could that be? And if you've watched this live stream already before, then I might have already talked about this also in the other YouTube videos that I made. Yeah, it's of course tissue. Yeah, it's not muscle. It's not muscle. Yeah, and it's actually something. Yeah, I'm gonna go up yet further. Sperm. Well, you're close. You're close. The sperm indeed can be seen here. They are in here. These are the so-called yeah. It's not a kidney. It's not a kidney. It is, I think, from a mouse. Yeah. And indeed, these are so-called the seminiferous tubules. Yeah. So it's in the testes. The testicles of a yeah. Not ovary. Not ovary. Okay, so these are testicles, and what you have in here is these are where the sperm cells are made. So in the cells, the sperm cells start to be made in the wall of the seminiferous tubule, and they differentiate, and as the cells divide, they move inwards, and then in the inside you have the finished sperm cell. Okay, so that is that's actually the way it looks like. Let me go up a little bit with. Yeah, I've also got other specimens where it's a little bit more clear. I think again it's cut a little bit too thinly, therefore you don't see it quite well. But in here, all of those little lines that you see here, these are the tails of the sperm cells. Okay. Yeah. So, but let's have a look again under the mobile phone. Okay, and let's see what it says here. Okay, so I have to take a picture again. Okay, it doesn't say which of which animal. Okay, just testes cross section. Let me try it again. Maybe when it retranslates the whole thing, maybe I get a different result. I don't know. Nope, it's blurry. I always have to focus. Again, it's again blurry. Again blurry. And see, it's maybe too close. It's problems. Yeah, it still says the same thing. Yeah, I don't know. Yeah, The source could be probably a mouse. Usually what they do is they use some animal, some non-human animal tissue simply because it's cheaper, okay, and more readily available. So what about this one over here? I don't know. It looks like a black square. Okay, and we're going to have a look at this now. Of course, I have to change over again. And let's see. Here it is. Yeah, this is so easy. But I think it's stained a little bit too darkly in my view. Of course, yes, it is a plant. And you know it's a plant because of the strongly visible cell walls that you have here. Okay. Yeah, so it's clearly a plant cell. It's one of the first specimens that you usually put under the microscope. Could this be the nuclei? I don't know. They seem to be. I don't think so, because look where they are. They're just scattered throughout. Normally the nuclei are a little bit different. Not so much on the top. Look. I don't think that look, this is way out of focus. These seem to be some kind of bubbles or so, because look, one cell normally has one nucleus, but look at this. This cannot be a nucleus. This must be some kind of a contamination here. Yeah. These are of course, yes, these are of course onion cells. Okay, stained onion cells. I will tell you the following. Staining doesn't really make a huge difference. I mean, everything looks now bluish green, but I think unstained you will see pretty much in the same quality. Yeah? So let's have a look again. Under the micro, yeah, look at this. All of those dots that you see here. One they look almost like oil droplets or something. Yeah. So I'm a little bit. I gotta be honest with you. I'm a little bit always afraid of wiping it, because if you just rub over it, then maybe you're just going to scratch off the cover glass, and I don't want that. So let's have another look here. What is this? Again, onion skin film loading. Again, film loading. I don't know what they have with this film loading. Okay, maybe hole mount. I don't know. Yeah, I don't know what this film loading always means. It has nothing to do with film, or maybe a thin cross section could also be. Yeah, still. Yeah, but we know it's an onion. Yeah, as expected. The last one here. Let's have a look at the last one. Go down again with the magnification a little bit, because it's easier for me to find whatever I want to look at. Here we go. Here seems to be something. Okay. Also seems to be some kind of tissue, obviously. Yeah. And let's open the condenser a little. It's kind of very blurry, yeah, but it's evidently stained. I think I know what it is. Yeah, it looks a little bit like muscle. Okay. One of the typical characteristics of muscle, we're going to check this, okay, is that the muscle fibers are actually very long, and they have multiple nuclei. The nuclei are stained here. Yeah? So that's one a very typical thing. It's one of the, so muscle fibers are quite an exception a little bit to how cells look like, because normally we have one cell with one nucleus, but then again here we have long cells that basically are fused together with multiple nuclei. But we're going to see whether this is indeed muscle. Okay, not that I'm saying something wrong here. At least it looks very similar to muscles. And let's have another look here. And you don't see this. Skeletal muscle cross section. Okay. Yep. Yeah. That's correct. Yeah, there are different. There are actually three types of muscles in the human body. One of them is the skeletal muscle. Yeah, the one that allows us to move. And then we have cardiac muscle. That's the heart muscle. And we also have smooth muscles, so which are not connected to bone. And those smooth muscles, they, for example, contract to move food along in the digestive system. Or the smooth muscles are also found in blood vessels to control blood pressure. So they're able to regulate the diameter of the blood vessels. Yeah. So that's basically five common slides here, which we were now able to identify. And what I'm going to do now is I'm going to go back again through the comments. I have to scroll all the way up again. Okay. And there's people already, talked here about bee legs. So much about AI translation. Yep, exactly. So I'm going to skip through the comments very quickly. Okay. The third leg of the bee, the one that collects pollen. I think that's the reason why one of the segments of the bee's leg was quite thick, because this is where the pollen is collected. Okay. Yeah. So comments, film loading is probably a badly translated for slide mount. Oh, that's an interesting one. Film loading, slide mount, film and slide. Like the slide of slide projectors, analog slides. That could be. That's interesting. Yeah. That's interesting. Okay. Yeah. Maybe this is where it came from. Can you check the translation one more time to see if it comes back with the same result? Yeah. Okay. Which one? That's the one. Yeah. I actually did check, and it sometimes does come back with the same result, sometimes not. Yeah. Okay. So yeah, so these are comments of the previous one. Could you then show a healthy human cells? Doesn't matter what kind of tissue, just for comparison's sake. I can do that. I'm gonna show you human cells, but honestly, we human beings have a couple of hundred different cell types. So it really depends which ones you want to look at. And if you look at human cells or animal cells in general, because they don't have a cell wall, but therefore it sometimes can be a little bit difficult to see or identify the individual cells. So for that reason, back in the day, after microscopes were invented, the biologists were very interested in observing plants, because the plant cell wall was so prominent, and they were very interested in the cell wall. And indeed, some biologists at that time, 19th century, were even wondering if the cell wall is not the living part of a living thing, and then the basically hole in the center. Now we know it's actually the other way around. The cell wall is just a non-living, so to say, outer part of a plant cell. Yeah? Besides the irregular shape of cells, overgrowth of individual cell population could be another indication of cancer growth and metastasis. Besides irregular cell shape, overgrowth of individual cell populations. It's an interesting one. Okay, so it's not only the irregular cell shape, but also when one cell type overgrows another one. Okay. Yeah, so I'm going to skip forward. How are tissues for humans prepared slides or animal slides acquired? Okay, that's okay. Yes. So that's with acquired. This could mean have a double meaning. You mean where can you buy them, or does it mean how do you make them? I'm going to talk about both of them. First of all, those commercially prepared slides can be bought, of course, online. Even over Amazon. Okay, so if you just want to get a cheap source of slides is Amazon. You just go and type in readily prepared slides, and that's actually one of those examples which I bought some time ago over Amazon, which is an assortment of different slides. So I'm not I'm not recommending or not recommending this here. It's simply bought this some time ago. There are many companies that have those, and essentially if you look through those specimens here, you're going to see that very often those slide boxes contain a total collection of a variety of different slides. Okay, plants and animals. So over here, for example, we have a top with plants. Down here, we have mostly animals, which we can look at. And those are fairly cheap. Now, I do also have other more expensive slides. And they're pretty old. It's labeled in German. Okay, typewriter. It's from the 1980s. And they're still really like new. Okay, even hand labeled over here. Okay, the quality of those is pretty good, and they're pretty expensive. No, not pretty expensive, but more expensive. And those slides can even be bought individually. Okay, So either in a slide box like this, or they also sell them individually. And the company also said is that if necessary, if you need to highly specialized slides, they even prepare them for you and they have to make them. So there is even some waiting time involved. And the reason why the companies make those is, of course, for medical training and for training purposes, right? And as references. Yeah. So those slide boxes, therefore, sometimes contain highly specific slides as well. Okay, and if you want, we can have a look at some of those here, right? So that's I mean, how do you make them? Basically, if you make tissue, then you have it's a lengthy process. I will tell you, I have not made it myself, animal tissue slides, because the process is extremely elaborate and lengthy. You have to dehydrate it, you have to stain it, of course, you have to embed it in paraffin, which takes time. Then you need a microtome to section it. You have to remove the paraffin with xylene, which is toxic. Not stuff that I generally want to do at home, right? So the preparation of animal tissue can take several days, and then it's a question of is it worth it, right? However, there are a whole range of methods and protocols available for this. So this is a standard procedure that has been done for many years now already, but it does take some time. Yeah. Would you have suggestions to remove ciliates from microalgae stain? I try to isolate in a pond. I tried alcohol, but it kills the algae as well. Okay, so that's a specific question. You want to remove the ciliates you want to get a pure culture. Okay, that's essentially what you want to do. And that's always a difficult process because if you've got one cell in there that's basically not belonging in there, then it might reproduce. So generally, and in microbiology, when you grow bacteria, you do that by growing the organisms on agar plates in colonies. Now you cannot do that with ciliates and other large organisms, right? So and because of the biodiversity that is there, how would you do that? Because one method might kill some ciliates but not others, right? So alcohol and other substances like this are very general, and they will kill pretty much all living things because alcohol dissolves the cell membranes, and the cell membranes can be found in all organisms. So essentially what I would suggest is to have to kind of keep on trying to reculture them. So when you have increased of the microalgae, then you take a small sample of those and you try to continue to grow them. So you take a small sample, hoping that you do not transfer any ciliates along, and then you kind of keep on reproducing those, and you keep on doing these purification steps over a longer time, and then you might ultimately end up with a sample that does not contain any ciliates. So there's another one. Strange. I would have thought a cross section would be long and thin, but not round. Yes, this is indeed something I need to use this here as an example. Okay, let's say that this is a stem of a plant, and when they talk about a cross section, then basically you're cutting it like this, and you're looking at it from the edge, right? So it's round, right? And the other thing would be called a longitudinal section if you were to cut it like this, right? And then you're looking at it from the side, right? So this is the reason why it looks round, is because you're looking at it basically, you've sectioned it like this, and you're looking at edge onwards. I was watching the old live on filters and oblique illumination. Did you try the radial 3D filter? I tried it and it gives me the same effect as oblique, but in words for the objectives and iris apertures. The old live on filters and oblique. Did you try the radial? Not yet. There's still so many projects that I have lined up. Okay, but I think that's one I did not try yet. Okay, I wanted to learn the reproduction of unicellular ciliates. It's a bit confusing. It's binary fission. I wanted to understand it. Okay, so cell division of is generally an important thing, and you know what? Because this was a question, and I'm going to look at some other slides as well, but because this is a question that is also considered important, I'm going to show you the following. Just going to, just for the fun of it, make a small space. Look, let's have a look. I have not looked at this for some time now. Okay, this here is a pond water sample where I added some milk powder. Okay, some powdered milk, condensed milk. Yeah, powdered milk as a food, and I fed it too much. Okay, I gave it too much food, and when you do that, then essentially yeah, there's plenty of food present, and they start to grow. However, bacteria also start to appear, and they will push down the concentration of the oxygen. Okay, so what I therefore do is I will take some samples right from the top here. Okay, a very small amount. I think it's way too concentrated. Yeah, cover glass goes on top. Okay, and I have to clean this here because it's I don't like it. You have to have stuff like this with bacteria and whatever unknown stuff floating. So I usually I clean it because if I start to puncture myself now, then I might get an infection. So you got to be careful with stuff like this. Always you'll see it's Paramecium and milk powder. So let's have a look here, and I'm going to talk a little bit about binary fission now. Essentially, it's mitosis. So let me have a look first. Maybe we're able to see. Or maybe they already all died because the culture has gone stationary. Yeah, this was there was more activity going on a few days ago. Yeah, because what happens is the following: they will start to grow, and they will start to multiply. No, there are still a few left. Nah, this is a bad one. Yeah. Yeah, and sometimes you are able to see a few of them dividing. So these seem to be some smaller ciliates here. And it works like this. When essentially the division process of ciliates follows the same basic rules as the cell division in human cells or in plant cells, and it's called mitosis. And there are indeed the same basic steps. Number one, the DNA has to be copied. Let's just say that, I don't know. I'm going to use this as an example. This permanent marker is a DNA. Then it's going to be copied. Okay. Then you have 2 copies of the DNA. It's actually not that simple as I'm saying, but you have 2 copies of DNA. Then within the cell, they're separated. Okay. The DNA has to be separated. You still have 1 cell. Yeah. And then as a last step, the cell has to divide. So this basically means that the cytoplasm is physically divided, and then you have 2 cells containing the same DNA. And this process of copying the DNA and separating the DNA and then dividing the cell is the same for all living things. In bacteria, in prokaryotes, how this happens is a little bit different than in eukaryotes, because in prokaryotes, in bacteria, and you're able to see a lot of these bacteria here in the background as these tiny little dots, which are almost very difficult to see. Most likely these are bacteria, not the larger circles that you see floating around here. These are most likely ciliates or flagellates, eukaryotes, but the bacteria much smaller. Those bacteria, essentially, they have much shorter DNA and a much simpler DNA, and therefore the DNA is separated differently. In eukaryotes, basically the cells, like in ciliates, what they have is they have several DNA pieces, and the chromosomes basically are those things here, and that's the DNA, and then separated, and then the cell divides. So I just want to say that it's in 3 minutes I explained now stuff that usually takes me I don't know, a couple of lessons in school to explain. Yeah. I'm going to collect a few bugs. Do you recommend keeping them in a bioethanol or isopropyl alcohol if you recommend one? It doesn't matter so much. The difference between isopropanol and ethanol is one difference, that isopropanol contains less water. It is drier. So if you want to, and isopropanol is, I think, also a little more expensive than ethanol. So I think ethyl alcohol is perfectly fine. It's around 96%. Yeah, and that's fine. Isopropanol is generally only used if you really want to completely remove all of the water for further processing. So for example, if you make tissues of animals, then it has to be completely free of water. And even the small amount of water that is in ethanol is too high. And therefore you then do it first in ethanol, then you transfer to isopropanol to remove the rest of the water. So to answer your question, isopropanol is also works, but is expensive and or more expensive, and therefore not necessary. Okay. By chance, did you read anything on the 5 moss and 6 fluorescent blue staining from crushed orange climber roots? Looks interesting. No, I didn't. Sorry, cannot tell you anything about here. Those stains. Okay. Yeah, the same slide box. Yeah. And how are pathology prepared slides made? Well, essentially, pathology prepared slides contain tissue specimens of various diseases, like, for example, tumors. And in hospitals, they frequently make those. Sometimes they even have their own microscopy department where they are prepared for microscopic diagnosis even. You take a tissue sample, and then what you have to do is you have to dehydrate it. So this basically means you put it into alcohol, increasing alcohol concentrations. And you're increasing it step by step so to not be too stressful on the tissue. Because if you drop the tissue directly into concentrated alcohol, then it's going to shrink too quickly. And so that's why you increase the alcohol concentration slowly to slowly remove the water. And then at the end, you put it into a substance called xylene, which is not healthy, to make sure that all of the water is gone and it's a solvent. And then you put it into paraffin, and paraffin is wax. And so it's embedded in the block of wax, and then you're slicing it with a microtome. There is also a staining process step involved. And then again, the paraffin, the wax has to be removed with xylene, and then you have you make a slide, a permanent slide. And this takes actually a couple of days to do. Okay. And how do algae reproduce? If they do, of course they have to reproduce because all living things have to reproduce, and it's the same way. There are cells in algae that divide. Okay, and the DNA has to copy, the DNA has to separate, and then the cell will divide. Yeah? So this is the basic principle in all living things. Any fun experiments you can suggest for using Gram's iodine for staining? Well, Gram staining is used for staining bacteria. I mean, I wouldn't say if there are any fun experiments, but staining is generally kills the organisms that you want to stain. So there is not a lot of movement. But sometimes what's really fun is to observe things that actually move. Okay, setting up DIC seems to be pretty expensive. Lately, I've heard about Christiansen illumination is cheap. I have never heard of this, at least not under this name. It's called Christiansen illumination. Okay, thank you very much for the suggestion. The only thing that that gives a similar image as DIC would actually be oblique illumination. Okay. So what I'm going to do now is, as promised, I'm going to have a look now at a few other specimens here. I just wanted to show you again. So I'm going to take out of the slide here. Just want to show you what I've got here. Up. Look. I'm going to switch to the desk again. It's a plastic box where I just drop in all of my used slides, and then yeah, later on I'm going to clean them. Actually, I'm going to wash them with some dishwashing powder, and I find dishwashing powder to be very effective. As a matter of fact, much better than soap. If you just put some dishwashing powder in some hot water and let it stand, the slides are going to be pretty pretty clean. I'm not going to wash them now, but later on. Yeah. So let's have a look at a few other slides here from the slide box. Okay, lots of it says here, yeah, for schools, right? Yeah, I'm going to now show you a paramecium, but a prepared one. Okay, so because it's also interesting. So stained and prepared paramecia which do not move anymore. Okay, normally I don't recommend that because I think that ciliates and paramecium are much more interesting and fun to observe when they are still moving. But over here, there are not a lot of them on here, but a few of them here. Okay, yeah. So let's have a look. Okay, because simply as a little bit of a comparison on what to expect. So I'm going to remove again DIC. I have to go down with the magnification, and yeah, here we already see a few of them. Those long things, structures that you see here are Paramecium, but it's simply too overexposed now. So we're going to, this does not really matter, so it's in bright field. Okay. And here we go. I think they must be stained. Unfortunately, the description does not say that. Okay, but there is one huge advantage of those prepared slides, and that is that it's now finally possible to actually observe a Paramecium relatively easily, because normally they move around too quickly. Okay, so it's kind of difficult to observe them. So let's go up 60 times. And this is how it looks like. Okay, and I guess maybe that this dark part here must be the nucleus, so maybe they used some stains that stain DNA. And if you actually focus through it, you're able to see the different vacuoles, maybe food vacuoles and vesicles and so on. Yeah, just wanted to show it to you because normally we're used to seeing Paramecium swim around. Okay. Safranin could be possible. Safranin is a red stain. Okay. So that's quite well possible. Okay. So that is basically simply a short comparison here, which shows you a little bit the advantages and disadvantages of permanent slides over wet mounts. Yeah, that is that it's possible to stain them. Of course, if you try a live stain on Paramecium, that's difficult. Okay. And a few other interesting things here. This is, I made also a YouTube short recently in my other channel. This is quite a nice one, and because some of you were asking about medically relevant slides, they actually also included one here. This is Trichinella. This is a parasite, which is a worm. It's a nematode worm, which basically forms a cyst in muscles. So if you eat, especially pork, which contains those cysts, and you eat them and the pork is not properly cooked, then you might actually also get those in your muscles. And I just want to show them to you because it's actually one of the nicer slides as well. I need to, of course, switch over. And we're able to see again here the muscle fibers. And this now actually shows you how nice muscle fibers can look like if properly prepared. You just remember a couple of minutes ago when you showed you the other one, where I made you guess what it is. And these look actually so much nicer. So, and let's have now a look, and I just want to explain to you. You see that the muscle fibers, some of them, they run in different directions here, but we're now going to try to hunt down a cyst. And I've got one already over here, and that's this one over here, and this one over here. So these are basically the cross sections of the nematode worms that have embedded inside the muscle. So I'm going to go up with the magnification again. Okay. And because they are, they kind of wound up in, and because it's a cross section, therefore it looks like this. So you see that you have over here 2 trichinella cysts in this meat here. Okay. There's the question is, what are the symptoms? Well, what I read is that, well, if there are not too many of them here, it's not a big deal. But if there are too many, of them, it can actually lead to muscle cramps. But those cysts can survive for a very long time. Because essentially they're just waiting there until the animal that has those cysts is eaten up by the next animal. So, for example, this is a problem if you have pigs, pork, and if you feed pork meat to porks. That's not a good idea. Also not allowed in some countries, simply because this way you're kind of closing the reproductive cycle of those trichinella worms. And generally, it's not a problem in these days so much in most countries, because the meat is checked and it's also not a problem if the meat is properly cooked. Yeah? But sometimes this is not the case, and then you have the problems of trichinella, trichinosis, I think. It's called. Yeah? But simply interesting to see that this general purpose slide box actually also contained these medically relevant slides here. Okay. Yeah. So I'm going to quickly read again some of the comments here. Okay. What's the difference between mitosis and meiosis? Mitosis is cell division in eukaryotes. In meiosis is the production of sperm and egg cells. So before that, when I showed you the cross-section of the testes, this is where meiosis happens, where basically sperm cells are produced. And the purpose of mitosis, of regular cell division, is that you make two cells that are identical, genetically identical. And in meiosis, you're making sperm and egg cells that are genetically different. Because that is the point of sexual reproduction, is to make sure that every generation is different from the previous generation. So for example, I'm of course genetically different from my parents, right? Yeah, I'm not a copy of my parents. Yeah, I'm a new individual, and that is the whole point. That essentially is a genetic change from generation to generation, so that the species is able to adapt to continually changing environment. So this is kind of the purpose of having sperm and egg cells that are different from each other. Okay. Can you go into horizontal gene transfer? How does this work? Are the microbes trading genes like baseball cards to gain new abilities and functions? Again, this is there are lots of biology questions here, and I think this would be a really thank you for those questions. But I think that indeed might make a separate video about these things, simply because they're quite interesting. Basically, it's the following: you have, in plants and animals and fungi, you have as I just mentioned, sexual reproduction, which makes sure that every generation is different from the previous generation. But what about bacteria? Bacteria, they only divide by dividing, right? The DNA is copied, and then you have two identical copies of a bacterial cell. But how do you actually make sure that there is also genetic variability? How do you also ensure that there's some kind of mixing of DNA happening? And one way is so-called horizontal gene transfer, which basically allows one bacterial cell to transfer their DNA to another cell. And if it is a useful property, then the cell will now is able to survive better. Yeah. If it's not a useful property, then it has a disadvantage because it's simply extra DNA, extra energy needed, and then it's not able to divide as quickly. So the bacteria have a system of trial and error. They're accepting new DNA. They're quite open to that. And then they're going to try, is it useful or is it not useful? Right? And so that is their strategy, or one of their strategies, at least, to ensure that there is genetic diversity happening. Right? This genetic horizontal gene transfer is, of course, also an issue, because if I have a genetically modified organism in a lab and it kind of is released into the environment, then this modified DNA can be passed on also to other organisms, and you don't want that, because you want to contain this DNA, right? Yeah. So are you gonna watch them now on the slides? Okay. So I'm quickly skipping ahead. A nematode worm, yes, those trichinellas are nematodes. Okay, as I mentioned before. So let's have a look. There is a product called ProtoSlow to slow down protists or better observe them, but I've never tried it. It's quite expensive. Yeah. Carolina Biological in the United States, which is an educational supplies company, they have ProtoSlow, and I only used it a long time ago at university. And what I do these days is, if I want to slow down the movement of a protozoa, is simply to use very little water. And by using very little water, you're kind of squeezing the Paramecium between cover glass and microscope slide, and this inhibits your movement. So, so this is Leberegel. I don't even know how to translate this into English. This is in the liver. It's some kind of a yeah, also a parasite that's able to live in the liver. Yeah, yeah. So here's the Latin name. I don't have to admit, I don't know much about the reproductive cycle of this one, but this one seems to be a parasite that somehow infects the liver, and it's a huge thing because it seems to be a yeah, a pretty large amount. Yeah. And that's, I don't know. Look at it. It's just it's the whole organism. Yeah. And it's almost like, yeah. I can imagine that those things that you see here might be parts of the digestive system. Okay, it's almost like a flatworm. Yeah. But it is a thin cross section, and I think one of the nice things about this specimen is that you can already see quite well that the individual cells are not easily visible because it's an animal, and animals do not have cell walls. I think this could be one of the reasons why, maybe, it's my hypothesis, the cell theory was first formulated with plants. All plants are made of cells. Okay. A guy called a German biologist called Schleiden. Matthias Schleiden was his name, and he formulated back in the day, 19th century, said all plants are made of cells. Yeah, and even Robert Hooke, who coined the term cell, he was able to see the cell wall of cork. But look at this here, this is an animal. Where in the world are the individual cells? Of course, I can zoom in, okay? But I'm not able to see where one cell stops and the other one starts. Yeah, so no wonder. Yeah, it took scientists a little bit of time to also discover that all animals are made of cells, because it's so difficult to see where the different cells are because they're so tight together, and the cell membrane that separates them is so thin that you are not able to resolve it with a light microscope. Okay, and so that is the thing. Yeah. So that's another parasite. And let me see over here in my other box here. Up, because I have got yet more. Let's look at some medically relevant or interesting thing. Ah, yeah. Sleeping sickness. Yeah. Trypanosoma. A parasite as well in the blood, which causes sleeping sickness. Okay. And then a very difficult one, malaria. I can also show that to you, but it's very difficult to actually find it. But this one here is quite easy to find. Yeah, so that is a sleeping sickness. So also parasite, which is also transmitted and which causes, if you look very carefully here, quite some changes in the blood. Where is it? Focus, 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 focus. Okay. And yeah. So let's have a look at a higher magnification. Okay, I need to refocus again a little bit. Okay. Where is the blood? Where are the red blood cells? You might wonder. We have to go up with the magnification even further. Okay. You know what? I'm going to put it in the filter here. No, not a good one. Okay, let's go to 60x, and now we're able to see already the following. Okay. Yeah, do you see those blue worm-like structures? These are individual cells, trypanosoma they're called, and these are basically the parasites that cause sleeping sickness. And if I refocus those circles that you see here, these are the red blood cells. So the trypanosomas, they reproduce in the blood, and yeah, must end up destroying the blood cells, and that's why the people become sleepy, because the blood is not able to transport oxygen anymore. So there, it's transmitted by the tsetse fly. When it bites you, then basically those the trypanosomas are transmitted as well. So, and that's one of again, one of these things. If you want to see those highly specific slides, then you basically have to obtain somewhere a commercial specimen. And I did make some videos on this as well. So these slides are pretty old already, so maybe, yeah, when they're new, maybe you're able to see them a little bit better, but I guess this must have been stained as well, because otherwise they wouldn't be so blue, right? Oh, and let's have another one. So malaria is going to be a really difficult one. And I'm not an expert on this here, okay, either. And because I did quite a bit of research on how to identify, because they're kind of small. And I have some problems distinguishing the malaria parasite from other debris and dirt and so on. I have yeah, and of course, again, what you see is you see the red blood cells. And then I did a little bit of research on this, and what you have to look out for is that sometimes in some red blood cells there is a blue dot or several dots. Okay, and these are the parasites that are able to that reproduce inside. Inside the red blood cells. And sometimes you can also see that some of the red blood cells actually popped open and released some of them. But you see those little dark dots here could be the parasite, but I mean, there's so many of them around that maybe it's also some kind of dirt. So it's not, for me, it's not quite clear. It's not a very clear thing for me to see, right? So this is something that I also wanted to mention, that essentially it is possible to do malaria diagnosis using a microscope, but proper staining it, then many diseases or most diseases you're not able to diagnose with a microscope, because you cannot see the parasite or the pathogen. But malaria is actually one of those examples where you can do that. But again, this is one of those cases where, I have had some problems. Maybe could it be that maybe those here? Do you see those? This aggregate of little structures here could be maybe something here. But again, I'm not an expert concerning this. But I've looked at some reference pictures online, and they actually showed and said that. You have to look at certain dots and certain patterns inside the cells. And sometimes there are those round half moon shaped structures visible, which basically I've not always been able to find in the cell. Okay. So, but I just also wanted to show it to you that those slides also exist. Okay. So I'm going to go quickly through here. I heard copper sulfate or copper acetate can slow down ciliates. Okay. Maybe. I don't know about those metals. I have to be honest with you, because sometimes metals like copper will actually also kill some of them. But it's all a question of concentration, of course. Mitosis. Okay. I think that's the most fascinating thing. I have so many questions regarding horizontal gene transfer. Thank you for touching on that. You know what? I've been thinking about doing some general biology lessons online anyway, but that would be rather a little bit more theoretical. Okay. My first water slide I tried, I had the perfect amount of water and had a ciliate standing still for. Oh, that's great. Yeah. Okay. Are tissues mounted like that considered living? Not anymore. If once they're mounted, they're not alive anymore, because they've lost all of the properties of life. They do not have a metabolism anymore. And that's why you're able to preserve them. So by completely dehydrating the tissue, you are essentially, of course, killing them. But that is necessary to make them, to preserve the structures. Okay. Malaria blood cells in the other pictures looks like they are a little spiky. Can you zoom in further at the dot inside the red blood cells to see if they're residue or real malaria? The thing is at the following, I cannot zoom in further because I was already at the maximum 60 times magnification, and the next higher one would have been the 100th times, and then I would have needed immersion oil. But I never ever put immersion oil on my microscope, on the permanent microscope slides. Okay. Okay. There are deer walking through my yard right now. Maybe you can somewhere find some fur of some deer and put it on the microscope. Yeah, can you explain in short what is DNA sequencing? Okay, I'll do that. because maybe I'm able to make a connection with microscopy a little bit. That would be kind of interesting. You know what? And simply because I want to quickly, where is actually? Yeah, I dropped it back. I just want to put some kind of a nice picture in it again. Yeah, I don't know. I'll choose anything. Number 25. Okay. Let me, and then I'll talk a little bit about the DNA sequencing. I don't know. It says here that these are supposed to be some bacteria, but nah, they're too small to see. DNA sequencing. Not just let me find something that some people have something to look at. Okay. While I quickly for 2 minutes explain DNA sequencing. That's already at the end. Yes, that's Volvox. Where is it? Where is it? Here it is. Volvox. Okay, that's Volvox is an algae, also a permanent mount. Okay. So let's say that I know that it is Volvox because how do I know that this is Volvox? Because I'm able under the microscope to recognize the shape. Okay, I see, I don't know, a pigeon, a bird somewhere. How do I know that it's a bird? Because I'm able to recognize the shape. Very obvious, right? And very straightforward. How do I know it's a dog? Because I see a dog, it has typical characteristics of a dog, and therefore I know what it is. And now we have the following problem when we deal with, let's say, bacteria or some other microorganisms, because I look at them under the microscope and they look like bacteria, and I know, these are bacteria by the general shape, by the size, and so on, but I don't know which ones they are. And now, how do I know that? Okay, how do I know if I have two bacteria under the microscope that kind of look the same? They're all, let's say, round. How do I now know that they're actually the same, or most likely they're not? And what you do is the following. And this is what I have also done, is you can now grow the bacteria and do a whole range of biochemical tests. And by which chemical substance is it made of? And you do a whole range of analytical chemistry, and then you're able, basically you take the bacterium apart into its chemical parts, and then you analyze what is there, and then you check with publications and with databases, and then you can narrow it down what it is, right? Because the microscopic view alone does not provide enough information. So that's one strategy. So microscopic view to identify only gets us that far. Number two, I can chemically start to separate it. Lots of time needed to do that. And then the third thing is DNA sequencing. And what this is the following, is that all living things are made of DNA. And the more similar the DNA sequence is, the more related the organisms are. Basically, humans, you and I, we have, I don't know, let's say 99.9% of DNA similarity because we are humans, right? We share all the DNA similarity with other living things, but at a much lower percentage. And what you do now is, and I have also done this with bacteria. I've grown the bacteria in liquid culture and on an agar plate. And what I've done is I've taken the bacteria, scratched them off from my agar plate, and I put them into a little bit of water. I suspended them in water, and then I cracked open the cells to release the DNA. Now, how do you crack open the cells of bacteria? Well, you take the little plastic tube and you throw it in liquid nitrogen. It shock freezes the bacteria, and the water expands inside the cells of the bacteria, and expanding water breaks open the cells, and then the cells pop open because of the freezing, and then the DNA is released. Okay? And then what I've done is I've taken this DNA, I've purified it, you add alcohol, you centrifuge, it takes a couple of hours, and then you take this little tube with pure DNA and you give it off to the guy in the next department on the next door, because he has a DNA sequencer. So in the lab, I ran over and I gave it here, please get this sequenced for me, please. Because you got to be an expert in how to do that. Yeah, so I myself did not do the DNA sequencing because it's simply too elaborate. It's and the equipment is expensive. But inside the lab, they had this service company that did the DNA sequencing, and then you get an email back with a DNA sequence of a certain part. Yeah, and then you copy paste this DNA sequence into the database, and then it will tell you what the organisms are with the most similar DNA. Okay, the whole thing takes a day maybe. Yeah, to get everything sequenced and so on. Yeah, so DNA sequencing allows you to identify organisms where you have no idea what it is. Yeah, and you don't need a microscope. Yeah? So PCR, yes, it's PCR. You're using PCR, of course, to copy the DNA, and then you use they use also a so-called Sanger's dideoxy method for sequencing. The guy who developed it got the Nobel Prize, of course. Yeah, so it's a standard method these days, which is highly automated. So it basically you put in the DNA and it runs through it and it gives you the result. Yeah? So why am I saying this, and what is the connection to microscopy? Because there is one important thing. I'm getting so many emails where people send me pictures and they ask me, what is this? Is this dangerous, or which bacteria are these? And I can tell you, you cannot say that simply by looking under the microscope, because the microscopes sometimes don't give you enough information for some cells. This here, I know that this is Volvox, yeah? But for some bacteria that might be floating around there, you don't have enough visual information to actually be able to identify them. So this was also a quick excursion into DNA sequencing because you wanted to know, and I know that this is might be a little bit specific for some people, but still I think it's kind of interesting. Yeah? So what else do I have here? What time is it, by the way? It's already 1 hour 15, so maybe in about 15 minutes I'm going to. Yeah, this is one that I like a lot. This is wood. Okay. Again, plants, so I have to put this back where it belongs. And I like this a lot because it looks so nice. Okay, and in different sections, cross sections and longitudinal sections and whatever. Okay, yeah, and so that is basically wood that has been cut in one direction, and same wood here again. Okay, so you see that the cells are running this way. So I guess this would be also the direction of water being transported, but over here on the other side, it's a different, and here, different way. And here you can see the different cells. And you go down with the magnification. Okay, you have to swing out the filter holder here. And you can even see the annual rings. Okay, so every year, there is one additional ring being deposited. And these are also, of course, cells and tubes that are running radially, so that water is also transported this way. So that is quite a nice, one of my favorite specimens, actually, because it kind of looks kind of nice. Is there yet another one? No. Let's go up a little bit again. And here you can already see that there is a certain thickness. So it's not quite as thin. And if you check and go all the way to the side, you still see that there's a little bit of dirt there, but it's not as bad as some of the other slides. Is DSC dark field or other techniques still useful for viewing prepared permanent slides like these, or are they more dependent on water under the cover glass? Okay, yeah, that's the question is now, this here is in bright field. Now let's have a look. Generally, I feel that DIC works best in water because, let's have a look. If I do it like this, it also works, but it depends a little bit on the specimen as well. And but sometimes, depending on the mounting medium, I don't get a very strong effect. I think it depends a little bit on the characteristics of the mounting medium as well. Okay. So this is now again in DIC, and which adds a little bit of color here. Okay. Sanger sequencing could be time consuming and expensive, despite it has been a good standard. the new sequencing technique, nanopore, starts to get more traction recently. Ah, interesting. Heard of it, but I don't know how nanopore works. Yeah. Hello, man. I want to buy a dark field filter for the Stellar One. Where can I buy them? Good question. Yeah, you can't because you make them yourself. I would say this is an interesting thing that I also do not fully understand. Is that why microscope companies do not provide those very simple accessories, which essentially cost nothing to make but would add a lot of value, like for example dark field patch stops. These microscopes have a filter holder, and why not just throw in, just to make the customer happy, a dark field patch stop, easy to make. You can use this to say that you even can do dark field, but for whatever reason microscope companies don't do that. It's something I myself don't fully understand, but then again, those dark field patch stops can be made quite easily. All you have to do is you have to take a clear piece of plastic and you have to make the center dark. You just have to make sure that the center is dark, that the central patch stop has the correct size, and you do that by trial and error. I did make several videos about this, okay? But indeed, with the exception of those very high-end companies like Olympus, for example, I remember that my Olympus microscope, my first one, I essentially bought myself a dark field patch stop, and the company provided it. It was made of metal, high quality. I still remember it wasn't even cheap, even though it was just a disc of metal, right? Came in a big box even when I mail ordered it many years ago, but it was provided. But many other companies these days simply don't provide them, and I don't understand why, because I think they're quite cheap to make, and there should not be a reason not to provide them. So if anyone's listening from a company, microscope company, then I want to give you this advice. Okay, I want to show you now Lilie Staubblätter. Okay, was this the one? Where is this? Just a second. I'm looking for another one here. Was this 49? Was this the one that I wanted to look at? I don't know. This is a lily. No, that's not the one that I wanted to show you. Yeah, these are basically also the pollen grains of a lily. Those things here are the pollen grains. I was actually looking for something else here. Just a second. Potato. Was this over here, the one? Yeah. That's the one, number 23. 23, here it is. I think that was the one. Yep. I'm going to show this to you because, again, some people asked about cell division. So that's 49. Always got to put them back, otherwise I'm not able to find the slides again. Yeah. So what you see over here is again, a plant cross section of a lily, which is a flower. Okay. This is basically where the ovules are, the egg cells. And all of those little dots that you see, these are the stained nuclei. And so again, we're able to see the individual cells here. Okay. And we're able to see the nuclei, and in those cells, let's make it a little bit brighter. And this must have been stained with carmine, I think, because carmine is a stain that reacts with DNA. And what you can do is you can go up with the magnification. And now we have to basically, I think I might have shown this to you already some time ago. You can look for some cells that divide. And I've already got one over here. This one here is in metaphase. What you're able to see is the individual chromosomes here. Okay. Make it a little bit brighter. Okay. So, yeah. So these are the nuclei in here. Yeah, all of those round structures. But sometimes when you see those little sausage-shaped structures here, these are chromosomes. So this cell over here is dividing. Yeah. So just wanted to show you also, again, as a nice example, how also here chromosomes are visible. How much more easily it is to see the individual cells in plants compared to animals. because of the highly pronounced cell wall. Yeah. One more question. Will the resolution of the displayed sample on a microscope change if I use a phone adapter to picture the image rather than a very expensive adapter on the third eye? That's a complex question. Because there's so many factors that play a role. Generally, if you connect a mobile phone on to an eyepiece directly, what you're going to get is you're going to get a full field of view. Okay, so that means you're going to see the full picture as you see it, and as you look through the microscope, you're able to get the full field of view. And then, of course, if you then zoom in a lot, then the image is going to become blurry because you zoom in digitally. Okay, and if you connect a camera now directly to the photo tube, then you're not going to get a full field of view, but you're able to only get a smaller section in the center, right? This means that you're already adding an additional magnification right away, which also might make the image appear to be a little bit blurry. So what I'm trying to say here is that there are other factors as well, and you cannot automatically say which one is going to give you a better result. I've had photo adapters for the photo tube where the photo adapter itself kind of degraded the image quality again so much that the image was not as good as taken through with a mobile phone camera through the eyepiece. Even that is possible. Yeah. So I would say there are so many other factors available that it's difficult to say a little bit. Generally, however, if you want to get the best result, and if everything is highly optimized and if the adapter is good and everything, then you're still going to get probably the best result by connecting directly a camera to the photo tube. If everything is ideal, also because it's a significantly more stable connection. It's also something that you have to keep in mind, the stability and convenience of use. Yeah? So in that sense, this is something that I also want to say is that there are multiple factors that play a role here. Okay, is the longitudinal wood section there where some bunches of cells at the right angles have forgotten the function? Anthony, oh, I also forgot it. These are basically the cells that are transporting the substances in and out. Yeah, also don't know the name anymore. It is slightly different as I compared the same images taken between cell phone and the mounted camera. Okay, reminded me about the digital zoom. My comparison was without digital zoom. I tell you the following. I once had a photo adapter, DSLR adapter, that are connected to my stereo microscope. My stereo microscope, not the compound one over here. And with a stereo microscope, I got a pretty bad image quality. I was really not happy. And I got a much better image quality with the mobile phone through the eyepieces. Yeah? For whatever reason. And I mounted the same bad DSLR adapter now to a compound microscope, and the result was quite good. So so that's one thing. If you use a USB camera connected to the photo tube and the sensor is very small, you're going to add a lot of signal to you've got a bad signal to noise ratio, and you get a lot of a blurry image because you get a lot of empty magnification again. So then it's not so good. But if the sensor is larger, then it's probably better. So it's very difficult for me to give a general recommendation. My overall suggestion is if you're going to buy a new microscope, try to get one with a photo tube because it simply opens more possibilities. It's convenience and so on, opens more possibilities for later, but many people don't have a photo tube. And if you actually optimize connecting a mobile phone to the eyepiece, then and you're really able to get a very good stable connection, then this also works extremely well. Okay, but it's more tedious to set it up. Okay, so in that sense, it's a little bit difficult for me to give a clear answer here. So that is a little bit the thing that I simply have to pass on. In my experience, yeah, there are trade-offs with all sorts, but you get a reasonably good result if provided that you actually get yourself a reasonably good mobile phone adapter, which makes sure that everything is very stable. Yeah? But it's not so nice to always having to realign everything, and so it's more time consuming. So yeah, sorry not to be able to give you a more concrete answer here about image comparison. I've seen both already. The best result you do get if you're able to directly project an image without a optics or into the sensor of a camera, I would say. Okay, there are a lot of to do with each plane within the light path. That could be one of the major factors in consideration of the quality of images. Yes. Okay. Do you know next week's topic? It's 1.5 hours already, and I'm also starting to get a little sore throat. No, I don't know next week's topic yet, but I'm open for suggestions. As a matter of fact, today I actually was thinking about actually also showing you some pollen from the dandelion under the microscope, and hopefully get some pollen tubes growing. But so far, no success. Yeah. But maybe I'm able to get this done. Optimized for next week. Diatoms. Yeah, sure. I've got some diatoms. Some samples which I can show you. Also some nice prepared diatoms, marine diatoms, which are around, and they also look quite nice. Okay. Water bears, also a possibility. Today I found a little bit of moss, so it's also a little bit of luck if I'm able to find some water bears in the moss. Eyelashes to move diatoms is a thing that requires a very calm hand. Never tried this, but you can actually take eyelashes, glue them on a stick, and you can actually arrange diatom lines. Okay, so never tried it, but I know that this has been done traditionally. Okay, so people, you know what? I'm going to call it quits again for today. Maybe water bears next week. I'll see. Okay, if I'm able to find a few of them. I think I'm going to call it quits for today again. Always open for suggestions and recommendations. Hope that you liked it, and I wish you a nice day, nice weekend. Still happy microbe hunting. And I don't know if you're celebrating May 1st. In some countries, it's also a holiday. Okay, and then you can use that time also to do a little bit of microscopy. Bye bye. See you around next week. All the best. Bye-bye.